It's my pleasure to introduce our last speaker for the session right before our panel discussion. Our speaker is Dr. Hiroki Ueda, team leader of Laboratory for Synthetic Biology at Riken Center in Tokyo. Welcome.
Can you hear me? No? Yes? Okay. So my name is Hiroki Ueda from Tokyo and also Osaka. So today I'm going to talk about the sleep and wake, especially sleep. So if you feel sleepy, that is not due to my talk is boring, but my topic is sleep. Actually, a little bit, irony because I'm going to talk about the sleep, but I have also sleepiness. I feel sleepiness because I took the overnight flight from Tokyo today, and then with jet lag and also sleep deprivation. Okay. Anyway, so recently, I didn't talk about the other technological work from our lab, but last year we published a paper on the whole brain profiling of every single cell existing in the mice brain, and then recently new atlas is accepted by Nature Protocol. So it's contained 100 million cells existing in the mice brain, okay, which might be useful for modeling actually. Our brain have a three state. Which is a wake state, and also non-REM sleep state, and REM state. And then transition among these three states is very important for the neurointelligence. And then the state of these, these states are very basic, so which give you some kind of a boundary condition for the very important function of the brain. For example, consciousness. Okay. But sleep and wake have some kind of balance, which we call homeostasis, and which is already known for many years, but nobody knows what's going on inside the brain or inside the neurons. Okay. For example, so if you have a long night, and next day you feel sleepy. Okay. So therefore, we think there is a homeostasis. Okay? Somehow, your neural activity will be counted, okay, and then converted to the sleepiness. And what is important here is the time scale differences. Neural activity is millisecond to second, and then sleepiness lasts more than hours, even days. Okay? And then somehow, some kind of a integrator counted up neural activity over time and then convert it to sleepiness, okay? And then suppress your neural activity, okay? And the question is, what is the sleepiness? Okay? What is the sleepiness counters? So this is a kind of a Japanese traditional, counter of the water, okay? To make a noise, to scare the deers, okay? And then there are two kind of completely opposite concept for the integrator of the time or sleepiness. Okay? So one example is like Japanese traditional water counters. And then you can imagine the water in the hot tub. Okay? So, and the hot tub in this case is cells. And then within the cell, amount of molecule is accumulated and they represent the time or sleepiness. And then this is easy to understand, very intuitive. But there is another concept. Okay? In this case, time is something like the a picture or drawing in the paper. Okay? So in this case, molecule itself is a kind of integrator. And then within the molecule, state of the molecule will represent the time. For example, modification of the protein like phosphorylation, methylation, or other modification may be able to represent the time. In other words, sleepiness. And then the question is, how our sleepiness is integrated and then represented in the neuron or in the brain? That's a question, okay? And the many studies are focusing on the sleep, okay? For example, in this case, during the wake, pyramidal neuron have a tonic firing, and then during the sleep, deep sleep, pyramidal neuron have a bursting behaviors. Bursting, resting, bursting, resting, bursting, resting. And then interestingly, frequency of this bursting is about 0.5 to 4 hertz, okay? And then over the time, and then macroscopically, slow wave activity, which is exactly matched with the 0.5 to 4 hertz, and then power of this frequency will be accumulated if you awake, and then further accumulated if you cannot sleep at all, and then go back to normal level if you have a sleep. Okay? So somehow macroscopic EEG data correlate with the microscopic bursting frequency. Okay? But the question is, how can we bridge the gap between these two, macroscopic and microscopic? And of course, one way is modeling. So since 2000, many people, many interesting and also pioneering work are focusing on to model these slow wave activities by using more than 1,000 neurons, and then succeeded to recapitulate the basic feature of slow wave oscillation. But model itself is large enough, quite large, and then computational time is limited. So therefore, these models are a little bit limited in prediction powers. So therefore, we try to take the kind of a, radical approach by using the mean field approximation to narrow down the model size. So in our case, in our model, we suppose average neurons are interacting with each other to generate the slow wave oscillation here. Okay? And then we suppose 20 million, we generated 20 million different models, selected more than 1,000 successful slow wave oscillation models, and then perform the bifurcation analysis to understand which proteins, which channel, or which pump are important to generate this one. Okay? And then we found just four common, channel or pump are important to generate these behaviors, which is turned out to be related to calcium. Okay? So during the bursting, calcium go into the cell through the voltage-gated calcium channel or NMDA receptors to stimulate the calcium-dependent potassium channel to decrease the membrane potential to suppress the firing. And then accumulated calcium go out through the calcium pump. Okay? So therefore, calcium suppression no longer working here, and then you can observe the bursting again. That's the mechanism underlying these models. Okay? So if this model is true, you can predict something. Okay? So if you impair the entry pathway or execution pathway of the calcium, you will expect more awake because calcium is difficult to accumulate. On the other hand, if you impair the exit pathway of the calcium, you will expect more sleep because calcium is easy to accumulate. Okay? So therefore, we try to test this hypothesis to knocking out the genes involved in these just four gene families. But we encountered a huge problem because in our genome, just 4 gene family consists of 29 genes. Okay? How can we do that? Okay. And then, but we decided to knock out them all. Okay? And then challenge is how can we make 29 knockout mice? Okay? So if you use traditional or conventional, knockout technique, you end up with 1 to 2 years just for one mice. okay? And then, but if you avoid the crossing, you can make a knockout mice within 3 months. 1 month for bone, 2 months for rays, okay? And then we started this project around 2010. It took 6 to 7 years, but we succeeded to generate the new method to make a knockout mice within 3 months, and then knock-in mice within 4 months, okay? So I just introduced a knockout mice technique called triple CRISPR. So you may know CRISPR, right? But if you use a high dose of the sgRNA, you always encounter the mosaic problem, which therefore, newborn mice cannot be used for the phenotyping. But if you combine 3 CRISPR together at a low dosage, like 1/3 of the dosage, and then you use 3 CRISPR together to keep the total concentration as same, you can increase the efficacy of the knockout in the newborn mice. So we surprised to find more than 95% of the newborn mice is a biallelic whole body knockout, okay? And the 80% of the genome are covered by triple set of CRISPR. And then 70% of the genes have 2 independent set of the triple CRISPRs. So our lab have every week lab meetings, but starting with the report on the gene of the week. So 2 genes knockout are reported every week, okay? And then this can be done just 1 person. So even even small lab can do such kind of things by using new technique, okay? And then we have now knockout mice. But how about phenotyping? Okay. So in sleep, mice don't like EEG, EMG recording. So therefore, they bite the electrode, they took off. So therefore, we usually perform the neurosurgery. Okay. And then it's time-consuming and also invasive, which is a little bit difficult to do a high-throughput way. So therefore, we decided to invent a non-invasive method to measure sleep-wake cycle. And the solution is respiration, okay? So we put the mice into the chambers, and then inside the chamber, we measure the pressure change by using the piezo sensors. And then by using this method, we can distinguish sleep and awake by measuring the respiration patterns. And then we designed the chamber by ourselves, and then we designed the rack, which hovering the 6x6, in total 36 chamber. And then we designed the facility ourselves, and then scale it up to measure the more than 936 mice per week, which is a kind of a world #1 throughput, okay? Which is located in Osaka, actually. And then we try to measure the, knockout mice phenotype by using this method. And then we found if we knock out input pathway or execution pathway, we observed less sleep. And then when we knocked out exit pathway, we observed more sleep. Okay? Here is the data. So as you can see here, sleep is significantly reduced here and here and here. And some genes are lethal. So therefore, in that case, we use a pharmacology to perturb the function of the genes. Okay. And then in the calcium pump, we also observe the significant increase of the sleep. But you may wonder, this is maybe due to the on-target effect of the CRISPR, or maybe off-target effect. So therefore, we design the CRISPR probe again, and then redo the knockout experiment again. Okay. And then we observe the exactly same magnitude and also exactly same direction of the phenotype. Okay. And then finally, you may wonder that is due to the respiration artifact. So we perform the EEG, EMG recording, which is time-consuming, but we confirmed the phenotype by using the golden standard technique like EEG, EMG. Okay, so far so good. Okay, But calcium is a positive ion, right? So therefore, usually it's correlated with the excitation of the neurons. Okay? But in our theory or models, we believe calcium promotes sleep, which is correct. Okay? That's the question. If our theory or model is correct, if you inhibit the entry pathway of the calcium, you will expect the excitation of the neurons. So therefore, we developed a whole brain cell profiling technique called cubic. And then we try to clear the mice brain and then look at the excitation of the brain after fixation of the mice. So we administrated MK801, which is an NMDA receptor inhibitors, and then look at the ARC expression in here. As we expected, mice brain is highly excited. Okay? And then we tried to analyze layer by layer, and then we found layer 4 to 6, 2, 3, and 1. All of them are highly excited after inhibition of the calcium entry. This data is important for us because we model the layer 4 to 6. Okay? And then this effect seems to be direct because C4 positive neuron always express the NMDA receptors. And then interestingly, excitatory neurons, glutaminergic neurons, and then inhibitory neuron, like a GABAergic neuron, both activated. Okay? And then, okay, so far so good. But how about the, sleepiness counters? Okay? Because calcium dynamics seems to be important, and then that calcium, so we thought calcium dynamics may be counted by somehow, like a protein, like a calcium-dependent kinase. And then we focus on the downstream kinase from the calcium. And then one obvious candidate, the CAM kinase 2. Okay? So CAM kinase 2 is a lethal in fly, so therefore a little bit difficult to test this hypothesis in fly. And also in mice, CAM kinase 2 alpha and beta have a problem in crossing. But by using our technique, we can bypass the crossing. So therefore, we decided to knock out the CAM kinase 2 family, okay, by using triple CRISPR. And then we found CAM kinase 2 alpha and beta have a sleep phenotype. And then which is confirmed by the second set of CRISPR and EEG-EMG. And then, so CAM kinase 2, alpha and beta, seems to be promoting the sleepiness. Okay. Actually, that is the first sleep-promoting kinase discovered in animals. Okay, so based on that, we propose calcium and also downstream kinase, like a CAM kinase 2, is involved in the sleep or sleepiness. Okay, so if this is true, maybe phosphorylation activity or phosphorylation status of downstream CAM kinase 2 may be able to represent sleepiness. Okay, and then interestingly, CAM kinase 2 have the autophosphorylation activity. So therefore we try to investigate the role of the autophosphorylation. Okay, but the problem is CAM kinase alpha have a 54 possible phosphorylation site, and then CAM kinase beta has a 69 possible phosphorylation site. So therefore, we decided to make every single phosphomimic mutant. Okay, and then here is a kind of a working progress type report. But we found some phosphomimic mutant increase sleep. Here is data. So when we overexpress wild type CAM kinase 2 alpha, sleep duration is not changed. Okay, but when we express the phosphomimic type CAM kinase 2 alpha, we observe the 3.5 hours longer sleep. And then when we kill the kinase activity of these CAM kinase alpha, these phenotypes are not observed. And then unphosphorated mimicking a mutant does not elongate the sleep duration. Meaning, status of the autophosphorylation status of CAM kinase to alpha may represent the sleep duration. Okay. All right. So in summary, so we found calcium dynamics or calcium-related channel pump seems to be very important to determine sleep. And then downstream kinase from calcium, such as CAM kinase to alpha or beta, seems to be important for sleepiness. Okay. All right. So therefore, we propose phosphorylation activity made of a calcium CAM kinase 2 pathway might be related to, or even causal mechanism of sleepiness. Okay. And then remaining question is, how can we can CAM kinase alpha beta interact with these channel of pump is not known. This is a kind of a future study. Okay. So by that, I'd like to thank our collaborators, and also I'd like to thank you because somehow you suppress your sleepiness. Okay, so thank you for your attention. Yeah.
We have time for a question or two. Right down here. There you go.
Have you had time to look to see whether any of your phenotypes have deficits in learning and other kinds of behaviors?
Yeah, actually, NMDA receptor is, of course, involved in the learning, but LTP or learning itself is a kind of a positive feedback, and then sleep itself is a negative feedback. So that is interesting because same molecule seems to be important for the positive feedback as well as the negative feedback. So to have some kind of balance. So.
Question up there, right there.
Could you please tell us a little bit more how you use the 20 million or 20 billion simulations in order to identify what the 4 critical parameters were that guided the rest of your work?
Yeah, so we first, generated the 20 million random, parameter set, and then we try to select the slow wave type solutions, numerical solutions. And then after that, we have a ensemble of the successful model, right? And then we try to perturb the every single channels. So and then try to look at which channel is important to generate a slow wave oscillation. So when we slightly change the conductance of each channel or pump, we found slow wave activity turn out to be changing to the tonic firing or even resting. And then we try to focusing on the channel which give rise both slow wave oscillation or tonic firing. Okay. Yeah.
That was consistent among all the 1,000 models or whatever that survived your screen.
Yeah.
That's pretty cool.
Yeah.
Thank you.
Okay. Thank you very much, and I'd like to thank you.